In this video, 3D images of bacterial cells in near-native states at molecular resolution are generated by electron cryotomography. This is accomplished by first plunge freezing intact cells, or alternatively high-pressure freezing and cryosectioning the cell pellets. A series of EM projection images are acquired and used to generate a 3D reconstruction, or tomogram, which is segmented and further computationally analyzed.
Hi, I'm Grant Jensen from the Division of Biology at Caltech, and this is Songye Chen, who's organized our presentation today. We're going to show you how to do electron cryotomography of bacterial cells. Typically, a single individual would do the entire procedure, but because our protocols have been developed through shared experience, we will include everybody who's presently working in the lab. In addition to Songye and me, on the video you will see Dobro, Ariane Briegel, Alasdair McDowall, Ladinsky, and Jian. Other lab members produced the computer screen captures and helped prepare the accompanying written protocol, including Beeby, Pilhofer, Jian, Zhuo, Jian, Elitza, Tocheva, and Dylan Morin.